Hi, everyone. Thank everyone for coming. Thank you, everyone in the WebEx link as well for joining us. My name is Gilberto. I'm the SS2000 product specialist in Yokogawa, North America. And I know we're all very excited today to hear Kyle Saunders' presentation. He's a PhD over at the University of Surrey, and they have been doing great use of the SS2000 over there in Surrey. Some very, very amazing research. We're going to talk a lot about what they've been doing over there. And I just wanted to briefly go over what the SS2000 can do so that you understand very well what I was talking about. So it'd be good to introduce you what the system can do. So, the SS2000 is a benchtop high-content imaging system that is equipped with Yokogawa's dual microlens spinning disk confocal technology. So essentially what it does in the imaging department is it captures great images in 3D and at a very, very high throughput pace. And we are equipped with a live cell technology. So other than doing tissues that are fixed or cells that are fixed, you can also image cells that are alive. Now the SS2000, different than any other high content imagers, it has a very, very unique feature in that it is capable of sampling individual cells or subcellular content from the sample that you're imaging and isolating them into PCR tubes, PCR plates, or 96-well plates for further analysis. So this is a very, very unique feature that when coupled with the imaging can lead you to some amazing experiments. So essentially, it is a dual purpose system. It is a high content imager, a great high content imager, but it is also a tool for taking your research into the single cell or subcellular level of granularity. So when I talk about, sample analysis after isolating the single cells in the subcellular compartments, what do I mean, right? So there are a few major applications that researchers have been using when they are able to isolate single cells or subcellular contents. One example is sequencing. So let's say you have a specific cell of interest or a specific subcellular compartment of interest, and you want to understand the genome or the transcriptome of that region of your sample. With the SS2000, you can image to help you find that region. For example, let's say you're looking for an enlarged mitochondria, right? With the SS2000's imaging capabilities, you can find that mitochondria either through fluorescence, through bright field, and then of course you can take it out and isolate for further analysis. You're also able to do mass spectrometry. So people that are researching proteomics, metabolomics, lipidomics, like it's the case of Kyle's team over in the University of Surrey. You're able to take single cell or subcellular samples and isolate them to run through liquid chromatography mass spectrometry workflow or something of the sort. And you're able to obtain, for example, the lipid profile of a single cell or of certain organelles and so forth. So these are very, very unique applications that the SS2000 enables. And with all that said, now for the main event of today's seminar, I'll let it for Kyle to start his presentation. Kyle, take it away.
All right. Well, good morning, everybody. It is a massive pleasure to be able to present to you today. So, thank you very much, Jiba, for the invitation and also for the fantastic introduction there. It's actually very useful that you have sort of had a brief introduction to the SS2000 and its capabilities before I start, because I plan to go way back to the beginning of our research just to sort of explain as to how the SS2000 has changed things for us. So my name's Kyle Saunders. I'm from the University of Surrey in the UK, and I'm going to be talking to you about single-cell lipidomics today. Some of the challenges associated with it, some of the successes that we've had, and what seems like a common case with single-cell work is more challenges shoveled on top. And, this is a fledgling technique, so there are certainly challenges still to overcome, but, we've got lots of great people working on the case. So to explain a little bit about who I am and, where my team comes from, the University of Surrey is right in the southeast of the UK. I'm not sure if my cursor's showing up there. The landscape and the countryside is beautiful, but the weather is awful. There's a mixed bag of fruit. But the seismic facility based at the University of Surrey is, it was established by Professor Melanie Bailey, who's my boss, and is just hiding in the middle of these photographs here. The facility was established as a sort of a national service for biology users to basically use the SS2000 to isolate single cells and subcellular compartments of their biological models, typically 2D culture. And then we're pretty agnostic as to the downstream analysis that's used afterwards. So, we invite users into our facility to isolate their material, and then it's up to them whether they'd like to look at genetics, transcripts, proteins, metabolites, and as the title of this talk gives away pretty strongly, lipid, which is what we're interested in, and what we offer is sort of an in-house solution. So that's what we've developed mostly. So just to explain where our journey begins with single-cell omics, the story really begins with drugs, as I'm sure many interesting stories do. My predecessor, Sir Holly May Lewis, was a PhD student before me, and she worked with nanocapillary sampling of pancreatic cancer cells in the pancreas. And you see an example of this. This is really a home-built system that utilizes glass nanocapillaries attached to a robotic arm on a microscope. I'll give you a better idea of what that system looks like in just a moment. But essentially, this is sort of like a prototype before the SS2000 came along. You would be essentially sat in front of the microscope under ambient conditions, and you have to control this robotic arm manually. So the sample preparation was quite extensive there. But once she was able to isolate these single cells into glass capillaries, she used liquid chromatography mass spectrometry and was able to not only detect but even quantify drugs in single cells. Lipids were seen as a part of that analysis, so it became the focus of my PhD to turn this into a fully fledged single-cell lipidomic methodology. The workflow, shown sort of quite low resolution here, is quite simple. So you expose a 2D culture of cells to whatever experimental conditions you're interested in, whether this is application of drugs, radiation, or chemotherapy. And then, once a certain amount of time has passed, you can take those cells out of the incubator and start your whole single cell isolation. So this applies to the SS2000 as well. So you isolate your single cells into your glass nanocapillaries, and then you backflush the glass capillaries with some lysis solvent, and then we personally just push the sample into LCMS files for our downstream analysis. So with this basic framework, you can, it's really just limited to your imagination in terms of what applications you can use this for. To give you an example, I'm just going to go through an experiment that I ran a couple of years ago that was published in about 2023. This was the application of chemotherapy to a pancreatic cancer cell line. We were quite interested to see how the lipid profiles would change in those two populations based on treatment with gemcitabine, as gemcitabine is pretty much the most common chemotherapeutic agent used for pancreas. So we had two parallel cultures, one of them dosed with gemcitabine, one's a control. And after 24 hours of exposure, we sampled from both populations, again, using the manual system. And you can see this is a pretty normal microscope here, open ambient conditions, with this very, very difficult to use 3D nanominiature that you would essentially have to drive to the cell surface for isolation. So once you've sampled those cells, and if you're quite well fed and well caffeinated, you could get about 30 to 40 using that system. You would essentially backfill the starting composition of our mobile phase for liquid chromatography. This is because we were doing lipidomics, so the mobile phase was sufficiently organic to start lysing the cell membrane, and we wanted the best chromatographic peak shape we could. So we used starting mobile phase there. We stored our tips at -80, and then we pushed them into LCMS files using a syringe driver with a lure adaptation there. So what does the data look like, and what sort of experimental conditions do you need to use for this? So once I get control of my screen, I can show you some of the chromatography that we were using. Those of you who are regular users of chromatography will probably be quite startled at some of the conditions here. So people were always quite surprised at the fact seeing lipids at the single cell level using analytical flow chromatography. So we're at 0.35 mil a minute there, which is relatively fast, considering a lot of people for single cell work will go down to nano flow. But we are still getting a reasonably extensive separation. So a 28-minute gradient per single cell. We're using a C30 stationary phase, so we want maximum retention of our quite non-polar lipids there. And we're doing so on a thermo-Orbitrap Q-Exactive Plus. So this is high-resolution mass spectrometry. So with regards to the settings that we'd use on the Q-Exactive Plus, we found that for maximum sensitivity, we had to keep single cells themselves, the actual samples, at full MS, just MS1. The reason for this is the Orbitrap, relative to other types of mass analyzers, has quite a long duty cycle. So when it came to introducing MSMS experiments in there as well, it lost sensitivity when we get down to the really single cell level. So to compensate for that, we performed our MSMS experiments on a bulk lipid extraction, so that we could then translate that back to the single cell samples by the chromatography attention time. So the data, if I get you to have a look at A, for example, figure A, this demonstrates why we had to go MS1 just on the cells, because if you do a cell dilution here with MSMS, you can see that even before reaching the single cell, you've got 45 cells in an injection, and you're already getting one MSMS validated lipid identification. So this demonstrated the need to use that bulk formed QC for our MSMS. But if we actually want to look at the identifications that we can get from a single cell, I want to turn your attention to figure B. So in this work, we categorize the identifications at 3 levels of confidence: low, medium, and high. So low being a feature that was detected by the mass spectrometry. It passed the relevant statistical checks, signal to noise, background correction, etc. But we couldn't match that identification to any database. So this is a low confidence identification that we typically have to ignore. And this is a significant problem with omics in general. There's a lot of matter that we haven't characterized and haven't added into the databases yet. It's referred to as biological. So for now, we have to ignore this data. So medium and high confidence identifications are what we are interested in at this stage, at least. So the medium confidence identifications are those lipids that we can accurately mass match to a database. We use lipid maps for this particular study, and we get a decent number there, on average about 200 per single cell. And then you can see that the scale of the y-axis actually shrinks considerably for the high confidence identifications that we were able to do MSMS validation of in the bulk and then relate that back to the single cell. And the reason for this disparity, there are quite a few explanations as to why there is this quite large gulf between the MSMS validated features in the bulk versus what you get in MS1 at the single cell level. So you can see in figures C and D the proportions of lipid classes expressed at the bulk cell extract level, and thousands of cells, versus a single cell sample. In terms of the lipids that are expressed, we get much higher levels of phosphatidylcholines at the bulk cell level, whereas we get a lot more triacylglyceride and ceramide per cell. So the reason there are lots of reasons for this. So there are different solvent extraction systems at play here. The single cells are lyzed in mobile phase, the starting composition when a cell extract was a pretty traditional Folk extract with methanol chlorophyll. The matrix effects between the two types of samples are going to be considerably different. You consider that there are thousands of cells being injected, or thousands of cells worth of lipids being injected. So in terms of ionization suppression, the matrix effects are going to be very different between those two dilutions there. And finally, one of the main reasons that we're interested in single cell analysis is because cells are inherently heterogeneous between each other. There's massive cell-to-cell heterogeneity in cells that are supposedly genetically identical. And this is something we'd like to use single cell analysis for. But if you do a bulk cell extraction, you're really only seeing the average of the entire cell population. So if you've got cells that are quite different, then you're going to see differences in the lipid profiles. So, although we can get some output from this work, we see already where the improvement can be made. So if we take those medium and high confidence identifications, put them through multivariate analysis, we see between the two populations of cells, the ones treated with gemcitabine in green and the control cells in red. If we do an unsupervised analysis, so principal components analysis, we actually get a pretty nice natural separation between the lipid profiles of those two populations of cells. So this was really encouraging to see. If we do a PLSDA, which is a bit more supervised, we actually tell the model which cells belong to which plants. Then, of course, we're going to get better separation in that case. But what's very useful about a PLSDA is we can generate a VIP score list of those lipids that are contributing most significantly to the separation. So we can see which we can see which species are sort of being upregulated in this case. And we saw most significantly the lysophosphatidylcholine cholines were contributing to the differences in these two populations. And this was actually seen by a completely different group working with tank 1 cells, but at the bulk level after gemcitabine treatment. And the fact that we were reflecting in our single cell data set what had been previously reported in the literature at this early stage in our single cell work was so encouraging, because every mass spectrometrist is scared that they're reporting these very low sensitivities noise, and this was sort of validation for us that we were headed in the right direction, at least in terms of the biological. But this work was luckily published alongside the optimization in analytical chemistry last year, so very grateful for that. But with all experiments in analytical chemistry, especially, there's always further optimization. I've highlighted some of those issues already. We couldn't take our MS directly on our single cell samples because of the sensitivity issue. So we had to rely on that parallel bulk, which didn't read all that as well as we'd like. The sampling using a manual system was very time consuming and required a skilled user, not to brag. And finally, those cells were sampled in the open air without any environmental control. So, there's quite a short time frame as to when you can actually get the sample without worrying about the ambient conditions playing a factor in the biological differences. I don't know why I've used this mysterious animation, because, you've all seen the system already, but, just a quick flare of the dramatics there. The Yokogawa Stallone SS2000 was introduced to our group. And I'm just going to go through a couple of the reasons why this has made such a difference to our analytical technique, because it is an absolute powerhouse when it comes to sampling single cells. So I've got a small animation here that demonstrates, much in the way that Deva's presentation did earlier, that you can sample single cells in relatively confluent areas, and there's pretty much minimal disruptions in the neighboring cells. So we've got the nanocapillary right in the middle here, and as it falls away, after having aspirated a cell into the tip, you can see that the neighbors are relatively unaffected. Okay, right. So, you can see this in even more detail now the attention's been called. You're going to get a very nice cell extraction in this video with a very clean spot where it isn't impacting the neighboring cells. So you get very clean sampling. But more importantly, these cells are live as they're being sampled. So they're in incubated conditions. There's control for CO2, humidity, temperature. So you don't have to worry about limiting your sampling to a very short window before you worry about the outside getting in. And, in order to actually sample that cell, once your parameters are set, all you need to do is click on the cell, and the system will essentially go and pick it up for you. So there's, in terms of, extensive training and preparation for cell sampling, that's no longer required with the system. But that's not the only boost in, our analytical prowess that we've received. We've also gained access to a very powerful mass spectrometer, the Xenotop 7600. So this is a mass spectrometer. It differs to the Orbitrap in that it's a time of flight mass spectrometer. So its duty cycle is much faster. But the difference with this particular mass spectrometer is what they call Xenotrap technology. So what essentially happens is that when you fragment your ions in MSMS, the Xenotrap will essentially condense those ions to a packet before delivering it to the detector, if I understand that correctly. So that allows a massive increase in your sensitivity for MS analysis. And that is really what you need for your confident lipid identifications. So if we have a look at some of the the method parameters using the Xenotop, you can see some other tricks we've thrown in there to improve the sensitivity of this. So rather than go for 350 microliters a minute, we've actually gone for 8. So we're now operating at microflow chromatography with a microflow column. Unfortunately, there are no C30 microflow columns on the market yet, but just wait a couple of years and you'll very likely see one. And you'll actually see that our runtime, instead of the 28 minutes that we were using at analytical flow, we've actually been able to reduce to 16 minutes. So although we're getting significantly smaller flow rates, we're actually increasing our throughput. So, a very powerful methodology that's been adapted here. And my colleague Johanna von Gerichten has been fantastic in helping to optimize this workflow. In terms of some of the data that you can pull out of this thing, it gets pretty extraordinary. So I'd like you to turn your attention to this bar chart at the top left. So these are single cells that have been injected onto the Xenotop after being sampled with the Yokogawa SS2000. And we're getting, on average, about 160, 150 MSMS validated lipid identifications per single cell, which at the time was pretty much unprecedented in terms of the levels of sensitivity. If you have a look at the PCA just below that, you'll see, one of the demonstrations as to, using this for single cell heterogeneity in lipid expression, because if you do replicate injections of your bulk, for example, they're just going to be technical replicates. So you see in the PCA that they cluster pretty much identically, whereas your single cell sample have this really nice spread demonstrating the cell-to-cell heterogeneity. So a really nice visual demonstration of why you might be working at the single cell level there. And interestingly, even with all of these MSMS validated lipids that come directly from our sample, we still see that difference between the bulk level, thousands of cells digestible at once versus this single cell extraction, where in this particular example, with quite high levels of PCs, cholines, and relatively lower levels of triglycerides there. So, there are inherent differences between these two types of samples, and really does pay dividends to work with both of them. So in terms of the biological relevance of what you can pull out with the methodology, can you see any differences? And, when you include the MS2, absolutely you can. At the single cell level here, we've got 3 different types. We've got 3 different cancer cell lines. We've got CHP1s, we've got PANC1s, and we've got ASPC1s. And those of you who have worked with these cell lines before know that PANC1s and ASPC1s are 2 different types of pancreatic cancer. And in fact, you can tell them apart just based on their lipid profile. It's separate quite nicely. Is another encouraging reflection of the actual biological reality there. And again, we are capable of getting a very, very respectable number of actually MSMS validated lipid identifications based on that sample material. So one of the really nice things about using the SS2000 is not only its sampling prowess, but the fact that it's a confocal microscope and a pretty powerful one. So we've been really interested in getting some fluorescent microscopy that we can then correlate with the downstream analysis afterwards. And one of the examples of us doing that, still with regards to the pancreatic cancer cell lines, is the cells respond to oxidative stress. So, the tumor microenvironment is going to endure some pretty severe oxidative stress, especially if you're going to be treating with chemotherapy or radiotherapy. So, as a sort of mimic of these conditions, we treated the cells with some hydrogen peroxide to induce that state of stress. And one of the responses that we were interested in looking at was monitoring lipid droplets. So looking at the number of lipid droplets and the size of them, because these are sort of very, very strongly implicated with resistance to chemotherapy and radiotherapy in cancer. So we were quite interested to see how these progressed sort of over time after exposure. So after sampling from these two populations with the single cell isolation, downstream mass spectrometry, we were able to see in the oxidatively stressed cells significant increases in the diacylglycerides and triacylglycerides between the two populations, and we saw a correlation with the increase in lipid droplet size. Now that really does make sense, because lipid droplets are cores of nucleolipids surrounded by a monolayer of phospholipids. So the fact that we're seeing an increase of these triacylglyceride is absolutely reflecting what we're seeing in the microscopy as well. This is obviously we are analytical chemists first and foremost. So I imagine those of you who are more biologically inclined will let your imaginations run wild, in terms of the sort of correlative measures you can draw between your microscopy and your downstream analysis. So the optimization work with regards to these microflow DDA experiments and the experiment I've just showed it's just been accepted by analytical chemistry. So the workflow here is going to be available for you to read into it in a bit more detail, I believe within the next week. So very exciting times for developing these single cell lipidomics platforms. So I'm just going to talk to you a little bit more about the future work. Some of the applications that we want to apply these workflows to. And, because we're working with cancer cell lines, the application of treatment is always what springs to mind as relevant, especially with regards to, for me, what's of interest is the radiation-induced bystander effect to treatment resistance. So to begin this sort of line of inquiry, I'd sort of posed a very quick sort of rough approach at this radiation bystander effect by looking at UV radiation first of all. Just because UV radiation, if you're in a chemistry lab, most people have a UV, right? So it's not quite as complicated to set up a sort of first-class experiment. And we've seen through the microscopy indeed that the size of the lipid droplets significantly increase after UV radiation as opposed to a control. And we wanted to see if what differences would produce in the lipid profiles afterwards. So just to quickly run you through this experiment, we really wanted to look at the bystander effect through, imposing our own spatial effect in the cell culture, sampling from those different areas, and then seeing if that's reflected in the downstream lipidomics. So in these tin culture dishes, what I've done was control and most were irradiated with UV through this aperture, which protected the cells on the exterior of the cell culture dish and just focused the radiation into a selected circular area in the middle. And these cells were sampled 24 hours after exposure from the three different areas: the control, the directly irradiated cells, and what I call the indirectly irradiated cells, to see if we could detect a difference in those three profiles. So indeed, we were able to differentiate the three different areas based solely on their lipid profiles. So what's very nice to see here is that the controls, which are in reddish pink, and the indirectly irradiated cells cluster quite similarly in regards to that second, which is what you'd actually kind of expect. They'd be closer together in terms of what they've experienced environmentally. They haven't had their direct irradiation by UV, so you would expect them to be close to the controls. And of course, the directly irradiated cells are separating from the other two quite clearly. So this was encouraging to see that we're actually able to detect the spatial differences based on what we've imposed just based on the lipid profiles there. But we're obviously going to need to increase the complexity and the sophistication of our application of radiation, because people aren't really going to be using UV radiation for pancreatic cancer anytime soon. I can appreciate it. So the treatment that we're focusing on at the moment are going to be X-ray and proton therapy based. We do have some quite nice solutions at the University of Surrey. So X-ray radiation cabinet, basically like an easy bake oven for deadly X-rays, which is very nice and simple to use. And being a part of the Ion Beam Centre at Surrey as well, we have access to proton therapy. So obviously, I've spoken about the radiation-induced bystander effect there. This is one of the most interesting things that we're personally vested in investigating further, especially with regards to changes in lipid profiles. We know that cells that have been directly irradiated will impact their neighbours that haven't been directly irradiated, by various signaling processes, and that's going to impact the gene expression, proliferation, and cell viability, and create some serious off-target effects. So that's something that we want to investigate in a bit more detail. And we'd very much like to use proton therapy first of all to investigate that effect. So actually applying the proton therapy to a cell population that you want to extract single cells from turns out to be quite challenging. So if you want to operate the beam in and sort of relevant energies to actually target single cells, you need to use a sample material that's going to allow the photon to actually penetrate through and hit the cells. We've got a horizontal beam, so the sample has to be turned horizontally. If you were just to put a cell culture flask or a petri dish the correct way up, then you'd have to build an entire particle accelerator tower to irradiate it from the bottom, which obviously we're not interested in doing. And finally, the cell system actually has to keep the cells alive and healthy. And the impact that you see on them has to be due to the protons. So this has required a little bit of tweaking in the lab before we've been able to start the proton therapy. So the solutions available to us are mostly based on a Leica imaging slide. I'll show you what that looked like in reality in a moment. So these Leica imaging slides are used quite frequently for microscopy. They have a 1.4 micron thick PET membrane sort of in the middle here. And there's a recess between the top and the bottom of the slot, which allows for about 800 microliters of cell culture media containing cell to actually fit in this little space here. So you can treat the surface of your PET. We use a gelatin coating to get your cells to here. And then here's where the two systems that we're currently assessing come into play. So commercially, you can buy these hybridization cover slips from Grace Biosystems, I think. Yep. So these are commercially available. They fit the Leica imaging slides quite well. They just stick on, and I'll show you what like in real life in a moment. But there's a bit of a limitation with those. You've got such a small amount of media, and what we were interested in doing is dosing our cells with proton beams and then doing quite a large time course study afterwards, so 24 or even 48 hours afterwards, to then pick out when we wanted to sample for mass spectrometry. So we needed to come up with a bit of a different solution that's going to allow the cells to have some gas exchange, because if you take this cover off for microscopy, for long-term imaging, everything will evaporate, and if you keep it on, everything will suffocate. So we've also come up with this foam build solution here, where we've 3D printed some walls, and we've basically affixed that onto a Leica imaging flow with a lid, which is a bit more like it's sort of a bath for the cells. So here's what those two solutions look like side by side. So you've obviously got this very easy, sticky solution here that doesn't work quite well in reality in this imaging slide versus this 3D printed solution. And you can see that we've actually managed to get something that works quite well. It can be inserted onto the proton beam horizontally once those cells adhere to the PET membrane. We can quickly dose with proton therapy, and then take it back up into the SS2000 for our long-term time course imaging. So the experiments that we're currently in the process of doing require this time course imaging. So if we direct our attention to these slides down here, these are the ones that are going to be dosed with proton irradiation. So the aim of the experiment really is to dose half of the slide should be quite confluently, filled with the cell culture, tank 1 still. And then we've got the radiation-induced bystander effect there because we've got this indirectly irradiated cells on the bottom. So the aim is really to, first of all, just to keep this as a live cell imaging experiment, do a time course over 48 hours, 24 to 48 hours to monitor the lipid droplet proliferation and lipid peroxidation. This is also keenly implicated in mechanisms of resistance. So those are the things we're interested in looking at. And then we'd like to use the imaging synergistically for sampling to look at that time course and then say, okay, it's at X time that we're interested. And this is where we see a big spike in lipid droplet proliferation, or this is where it starts to plateau. Let's sample here. So it's using this long time course imaging really for making an informative decision on when to sample. And then of course we'd like to repeat the proton irradiation for our downstream single cell lipidomics and bulk lipidomics. So this is pretty much a drop in the ocean in terms of the experiment seismic team are currently involved in, and we're working on sort of bystander effects of viral infection, people looking at metals in single cells. So this is only a few of the applications that we've been working on. And if anyone's interested in any of the other work that we're doing over at Seismics, I'm more than happy to speak in more detail and of course get you in touch with other members of the team. But I've come to the natural conclusion of this presentation. So first of all, I'd like to give thanks to everyone over at the Seismic team. It's a great team to be working with, very supportive. I'd like to thank Jiba and Yokogawa once more just for inviting me to speak here. And finally, thank you all very much for listening and hopefully paying attention.
And we will now open the floor to any questions that the audience might have. So everyone, please feel free to type your questions in the chat box. It should be on your right, and Kyle and I can take the questions that you have.
Hello, is my microphone working?
Hey, Kyle, can you hear me?
Yeah, I can hear you. Is it? I think I'm looking at.
Just got our first questions here in the Q&A.
Yeah.
So Esther asks you, do you worry about fluorescent dye interference?
Yeah, absolutely something we're interested in. And this has become a question for us that's fairly new, actually, because, it hasn't been common for people to do mass spectrometry directly on cells that have been dyed with fluorescence. I mean, typically, if you're going to do bulk lipidomics, you do your fluorescence dyeing separately to any bulk lipidomics work that you would do. So this is I've got the raw files for a direct dyed versus undyed experiment using some of these lipid droplet dyes, Bodip and Nile Red, because it's currently is a question that we're looking to answer in a publication that we're currently writing. So I hope to have an answer in publication very soon. But something that's absolutely on the radar, and something that needs to be answered quite soon for this sort of direct mass spectrometry coupled to fluorescence work.
Awesome, thank you. And another question from Julie: how realistic and reliable is it to
obtain mass spectrometry data with such little sample as just a single cell?
Yeah, I mean, it's certainly it's only something that has been possible with the recent advances in mass spectrometry. So traditionally, if you look at sort of single cell transcriptomics and genomics, they've always been able to use PCR technology to amplify their material. Whereas you see here, it's the same problem with single cell proteomics. We're sort of stuck with the material that we have. There's no way to amplify our signal. So I think it absolutely is realistic and reliable to obtain that from a single cell because we have, those technological advances. And if we point to what was in this presentation, I think what has been really helpful for using the xenotoph method there is the ion trapping. So it, confers really significant boost in the sensitivity of your fragmentation afterwards, allowing you to look at that material at the single cell level. And, I've seen similar advances from, Bruker instruments as well, thermo instruments. We absolutely are now seeing the mass spectrometry catch up with what they're able to demonstrate, compared to that traditional single cell transcriptomics and genomics work.
Awesome. And then Dan asks, have you used the SS2000 to sample different types of cells, or is the SS2000 limited to specific cell lines?
Yeah, so one of the really nice things about, being at establishing this seismic facility is that we are getting quite a lot of interest now from, a lot of different biologists, and every biologist has their favorite cell line that they like to work on. They've all got different questions in mind, which means that we do need to apply this to a wide variety of cells. So I would say that this is most suitable for adherent cell lines. So I think that's what they have to have in common. So if your cell line is, just going to be sort of non-adherent floating, then perhaps, other single cell sampling workflows are better. But, Jiba, I know that you've sampled a good variety of different cell lines yourself as well. Would you be able to speak to that as well?
Right. So as I guess product specialist here for the SS2000, I can help with that. So no, the SS2000 is not limited to specific cell lines. And Kyle, you were perfectly right. We just need the cells to be adherent, and then we should be able to at least attempt to sample the cells or subcellular content. So there's no such limitation to one or two specific cell lines. Moving forward, Michelle asks, how many cells have you observed or are planning to observe to be able to consider significant differences between irradiated and non-irradiated cells?
This is a brilliant question, and one that is always on the mind when we're doing this very sort of selective single cell sampling, considering throughput. Because absolutely, from a biological perspective, if I go back to compare it to single cell transcriptomics or genomics, some of those analyses are using over 1 million cells, and therefore the statistics are really on your side in terms of just like the statistical power. So in terms of the cell numbers that we're looking for, so I'd say in terms of the amount of picking that you can get done on one day, for an experiment, I would say about 60 to 70. If you use the machine learning algorithms on the SS2000 to automatically, just sort of autonomously detect and pick up the cells, you could obviously leave it going for longer without much sort of user input, and potentially you could increase your throughput that way. But there are single cell systems that obviously work at that higher throughput, non-spatially, of course, if you are just flowing those cells. And I think if we try to compare to the sort of million cell throughput systems, then it's sort of unrealistic. So I think being able to use it for these very specific spatial experiments where you need to really zoom into the detail of what's happening whether that's going to be cell-to-cell communication or bystander effect here. I think that's where the sort of the desire for a system like this comes in. But to answer the question in terms of how many we're planning for the paper, looking at the radiation-induced bystander effect, it's going to be about 15 to 20 cells in each condition there. So relatively no end number. But some of the mass spectrometry data we're seeing out of that is still quite compelling, even with a low end number.
Great. And Michelle, in that question, he also mentioned looking at the RIBE, which I imagine is the radiation-induced bystander effect. Kyle, if you can comment on that too.
Yeah, sure. So I'm not in the presentation that was just shown to you, but as a matter of publication that we're working on at the moment with X-ray radiation and similarly the radiation-induced bystander effect. That is something that the early stages of that work we are seeing. We are seeing some pretty interesting effects in terms of the behaviour of polyunsaturated fatty acids, which we would expect to be impacted by radiation and oxidative stress from that. Receiving the dosage of radiation. So we are actually seeing it at the single cell level as well, pertaining to X-rays, which I did touch on at the end of this presentation because it's going to be a lot more clinically relevant than say UV radiation. So something we are seeing in the populations that we're analysing as well.
Very cool. Thank you, Kyle. Another question from Dan. If the SS2000 is able to perform both single cell and subcellular sampling, are there any planned experiments that involve subcellular sampling? What would be their biological significance?
Another great question, actually. There was a paper published recently, not by us. I can't remember the group exactly, but they were looking at lipidomics performed on mitochondria, and it was bulk still. But they had separated the mitochondria out quite robustly with ultracentrifugation, and they found that the lipid profiles for mitochondria were actually very different to the whole cell, where they tried to do whole cell bulk lipidomics. And I think that the difference in those profiles really reflects the need to go subcellular, because locally, we can have very different lipid profiles. And it is absolutely something that we are planning in the future. We've got lots of biologists coming in to the seismic facility, and they've all got their questions in mind, and a lot of them are subcellular actually. So we are currently working on protocols for picking mitochondria. We're currently looking at protocols for just isolating the nucleus for sort of cancer therapy delivery. That sort of thing. So it genuinely is a question of imagination when it comes to what would be interesting to sample and sort of how you would impose your own experimental conditions. So definitely something we're interested in doing.
So now a question from Adrian. Interesting talk. I think I missed it. For the lipidomic analysis, you recover the whole cell, or do you sample part of the contents and the cell can be kept alive in culture?
So the cells are kept alive during sampling. So the SS2000 is, it's a live cell imaging system. So where the cells are kept during imaging, it's got incubation, humidity control, CO2 control. So they are alive at the point of sampling. We isolate the whole cell using the nanocapillary sampling. So we take the entire cell into the capillary, and then we immediately transfer that capillary to dry ice to sort of try and quench the metabolism as quickly as possible. So it's alive in culture until the point of sampling. So we get because if you look at sort of more intensive sample prep work, for sort of mass spectrometry imaging, like MALDI or something, the cells are fixed. And you're not really getting that information you're not really getting that information from the live cell, which we're trying to observe or sort of preserve as much as possible.
I can contribute to this one here too, Adrian, just to clarify. The SS2000, like Kyle said, does have an incubator which can keep the cells in culture, which you are going to be imaging and sampling from, alive for long periods of time. Now, if you're going to sample subcellularly, so if you're going to use the SS2000 to go inside of the cell that is alive at the time of sampling and sample any subcellular material, you're going to kill the cell in the process as well. So just to clarify, the SS2000 cannot keep the cells alive post-sampling, if that's what you asked. But as Yokogawa, we do have another system, the SU10, which can perform subcellular injection and sampling and maintain cell viability. So if you're interested in that, I would look into the SU10. And that was the last question that we had here in the Q&A. I'm going to give it a few more instances to see if anybody has any other questions. But if not, we will end the webinar at this time. So we would like to thank everyone for their time and for their attention. If anyone has any further questions for Kyle or I, there should be a box located on the right side with our email addresses. You can feel free to reach out. This webinar will also be available on demand on Yokogawa's website if you'd like to access it later or to share it with any of your colleagues. So I'm going to end the webinar right here. Thanks, everyone, and feel free to reach out for any additional questions. Thank you.
That's great. Thanks a lot, Jeeva.
Thank you again, Kyle.